Hello, in this video we will discuss about the sodium physiology. So basically sodium is important in our body. Why? We will discuss. So the normal blood level is basically 135 to 145 milli equivalent per liter is the sodium, and the hyponatremia occurs when less than 135 milli equivalent per liter will be decrease the sodium to cause a hyponatremia. So intracellular is the 12 millimole per liter, while the extracellular sodium in our body is the 140 millimole per equivalent per liter. So when we will put the salt and we will eat the food, that food will be digested in the stomach and increase the lumen by several mechanisms co-transport with sugar and amino acid as well as hydrogen ion, which that is lead to absorb the sodium. So we will discuss about this mechanism. So first of all, we should understand about the overview of the digestive system and digestion of the carbs and protein, because the carbs and protein is important for the absorption of the sodium. So here is the stomach, which does contain duodenum, and the pancreas are present in the duodenum side of the duodenum. So when we will eat the salt containing protein and carbohydrates, so the carbs digest partially in the oral cavity due to the salivary gland, which produce the amylase enzyme and other enzyme, hydrolytic enzyme, which that is semi-digestion of the carbs, but protein will not digest in the oral cavity. So it will move into the stomach and digest into the stomach. That digestion is partial digestion, which that is long polypeptide. Protein will convert into the long chain of polypeptides. So after this, the protein digestion, the semi-digestion will begin to duodenum digestion. So the polypeptides and oligopeptides and oligosaccharides will be digested into the duodenum, and the complete digestion occur in the small intestine. So let's begin to understand first of all the stomach, which that is produce the hydrochloric acid and the pepsinogen from the parietal cell and chief cell, which that is produce two substance, hydrochloric acid and pepsinogen. This hydrochloric acid with pepsinogen stimulate by the hydrochloric acid to pepsin. This is the inactive to active form for digestion of the protein. So here I am drawing the small intestine, which contain a lumen, and what will be happen we will discuss. So here is the enterocytes. Enterocytes, also known as the brush border cell, which contain a brush border enzyme. And enterocytes important character is contain a microvilli and villi, which that is converted into the microvilli for increasing the surface area for digestion. So here is the oligopeptides and polypeptides are present, which that is a semi-digestion after the stomach. After that semi-digestion go into the duodenum. So here is the pancreas, which that is produce the trypsinogen, and this trypsinogen will convert into the enteropeptidase to conversion of the trypsin. And this enteropeptidase is important for the polypeptide to inside between the polypeptide, the breakdown of the peptide bond. So this here is the chymotrypsinogen will be converted into the chymotrypsin in the action of the trypsin. Trypsin is important for the conversion of the inactive chymotrypsinogen into the chymotrypsin. And this chymotrypsin is very important for the oligopeptides will convert into the dipeptides and tripeptides. And this basically dipeptides and tripeptides made up of amino acid. So these 2 amino acid and 1 amino acid, 3 amino acid. So after this move into with the sodium co-transport, so it means the sodium is moving inside due to the amino acid movement inside. So important thing is that tripeptidase and dipeptidase is a brush border enzyme will convert into the tripeptide into the dipeptide and amino acid single. So the sodium will move inside and into the interstitial space. So the basically the extracellular fluid, which that is allowed to move the sodium inside with the exchange of the potassium by using ATPase pump. So here another mechanism is also discovered here is that the sodium will move inside with the co-transport with the inside movement of the chloride as well as the hydrogen ion. While on the other hand, the more important thing is the hydrogen ion or proton and sodium co-transport basically is the ATPase pump used for the sodium exchange. And here is the proton will convert by the bicarbonate to carbonic acid if the acidic medium is decreased, so that is why, otherwise it will be not. So it will be dissociate if the acidic medium increase, so convert water and gas carbon dioxide, which that it diffuse directly, and the aquaporin is allowed to move the water inside. So here is the between the intercellular space, which that is also important for the movement of water from lumen to the inside of the blood circulatory system. While on the other hand, the sodium will also move by the co-transport of the sugar. So let's learn a bit more about when move into the circulatory system. So here is the blood vessel. This artery is also known as the mesenteric artery. We can say that is the gastrointestinal artery. So the sodium move into the blood. So the blood serum increase sodium. So the extracellular fluid is also contain 140 millimole per liter sodium, while inside the blood will be arise the 150 to 140, basically the 140 to 150 millimole per liter. So it means the blood serum sodium increase, and that means that we need the normal level of the sodium. So what will be happen? It will not move into the cell because the cell is also required the 12 millimole per liter. So it will be excrete from the nephron, from the urinary system or excretory system. So here is the afferent arteriole, which that is moved to the glomerulus, and the efferent arteriole, which that is used for the reabsorption. So the glomerulus and Bowman capsule here, this glomerulus is used for the filtration, so the glomerular filtration rate will be increased, and this glomerular filtration rate will be inhibit the renin production. So what is the renin? We will discuss also. Renin is increase the reabsorption by the constriction of the blood vessel. So here is the proximal convoluted tubule, loop of Henle, and distal convoluted tubule and collecting duct. So here is the vasa recta, which that is the efferent arteriole wrapping around the loop of Henle to make a vasa recta for reabsorption. So important thing is that during the high level of the sodium in our blood to decrease the reabsorption. Why? Because the sodium our bodies need to excrete extra sodium. So here is the inhibition of sodium absorb from the sodium potassium ATPase pump exchange. So this will be inhibited. Why? Because the sodium will remain into the nephron. While on the other hand, here is the adrenal gland, which that is produce the aldosterone. So it will be stimulated by the angiotensin system, which that ACE enzyme basically will not produce more. It means the angiotensin converting enzyme ACE will not stimulate the angiotensin 2 from the angiotensin 1. So this angiotensin 2 is special for the a stimulator of the aldosterone. So aldosterone will be inhibited by this mechanism. So the aldosterone will be also inhibit the aquaporin channel from the distal convoluted tubule. To reabsorption of the water will be not possible. Why? Because the aquaporin channel will be inhibited by the decreasing of the aldosterone. So important thing is that here is the sodium will directly move, is the passive efflux is also inhibited, and on the other hand the vasa recta will not reabsorb the sodium, so water will be excreted due to the aldosterone deficiency due to the angiotensin converting enzyme is not working. So on the other hand the sodium increase in the collecting duct, and the collecting duct will be excrete with the water to excretion more of the sodium. So that means the normal homeostatic function for the balance of the salt during the eating of more salt will be excrete through our excretory system. So let's begin to understand the sodium function. So the extracellular function and intracellular function we will discuss here. So the extracellular function is the conduction of impulse and in blood osmolality. That is the extracellular function. While the intracellular function is the water balance in the cell, and the concentration gradient will be produced. So the first of all intracellular basically water balance and concentration gradient, and the sodium will be more, so the water will be more. It means the water retention in the cell, and the sodium will be less, the water will be less, and the concentration gradient mean the active transport and symport ions will be occur due to the sodium in the intracellular. So on the other hand the extracellular mechanism is their function is the conduction of impulse and in blood osmolality. It means the blood osmolality means the increase the sodium, it means the increase water retention. So the conduction of impulse means the sodium potassium exchange in the nerve. So the blood in blood osmolality means the blood volume will be regulate and blood pressure will be regulate, and the water retention increase to lead increase the blood pressure. So on the other hand the conduction of impulse is the peripheral nervous system, and the central nervous system will lead to conduction due to the sodium potassium exchange. So it means the sodium is very important for the nerve impulse and osmolality and homeostasis. So I hope you make sense. Please make sure to subscribe, like and share this video.